Let's see how far it goes, and if not, I can. Uh-huh. Well, someone's coming out from the back, so we'll see how we go. So what I'd like to talk to you about today is continue on this theme about morphogenesis and cell migration. And I think from the embryo, you've seen from Keese's work and Sean and others that we really have a lot of fun looking in vivo as to what's going on. And this pattern of cell migration here is really neat because you can see it's not an uncontrolled invasion into the microenvironment. These cells are following pathways that are stereotypical. There's a bunch of cells out in the front here that seem to be leading the way, and other cells coming behind. And so what I want to talk about today is some really neat observations we made, and then trying to put that together with some molecular correlates and some computational modeling to link this in. And I've been spending a fair amount of time reading about animal migrations, and John had mentioned in the beginning with the introductions that he likes animals, plants, cells, everything, right? So I think that there's a fair amount of overlap. And Hugh Dingle, who is an ecologist at UC Davis, had an article in National Geographic a few years ago, and he came up with, or sort of brought forth, this idea that there's 5 features of long-distance migrations that are persistence, linearity, undistractability, special behaviors, and stored energy that seem to be part of phenomena of just about every really animal migration, from ants and crabs and zebra, etc. So I started to look at this a little bit more, and this is a beautiful animal. It's the fastest land animal in North America, called the pronghorn. It undergoes 125-mile bidirectional migration every year in Wyoming. And what's cool about this is you've got these pronghorn going across this migratory pathway. Even though there's construction at certain sites, they've got to close the freeway down because they cross at a certain location. And it's just amazing. The problem, though, is, even though maybe Hugh Dingle would argue, is it's difficult to ask this antelope, what special skills do you have that make you a good leader? Is it experience? Is it a keen olfactory system? Do they have the smells of that microenvironment and that trail? Is it keen eyesight? We know that the pronghorn can have this visibility to detect, very long range, and they can spot motion and they can spot danger much farther out than most animals. The problem is that you can't ask these animals the questions that you want, except maybe Doolittle could, right? But we don't have access to Doolittle, and as much as I tried this summer to communicate with this dolphin, the dolphin didn't respond to me. So I'm going back to the cells. And I'm asking the question then, how do cells interpret microenvironmental signals? Because I can interrogate them if I can visualize them. And how do they move in a directed manner then? And the second question is, if they're responding to microenvironmental signals to move in a directed manner, then how is it that they're communicating this information to move as an integrated population? Okay, so those are the 2 questions I want to ask. In my model system, going back to the cells you saw in the beginning, are the neural crest cells in the embryo. They're very highly invasive. They undergo these stereotypical pathways that are accessible to in vivo imaging in many embryo model systems, and they form these beautiful discrete streams. They're an important cell population because they're multipotent. They give rise to neurons and glia of the cranial ganglia in the head. They form almost all of the cartilage and bone of the face and neck and connective tissue. So they play a major role, and if they fail to differentiate properly or fail to migrate, then you have a whole huge class of birth defects called neural crestopathies that are related to the neural crest. And since they're very highly invasive, they're also a model for neural crest cancers that are neuroblastoma and melanoma that are very, very invasive and can be very aggressive. Now, versus the zebrafish, I want to go back to the avian model that Keith talked about yesterday. We use the chick embryo model in the quail by cutting a hole in the eggshell and putting in a Teflon membrane that's oxygen permeable, but it's liquid impermeable. So you can keep the contents of the egg moist for up to 5 days and visualize the embryo, or you can pull it outside of the egg and then put it on a microscope stage. And we've also been mixing in different DNA plasmids, as Sean mentioned the power of multicolor labeling, to be able to refine processes between cell neighbors and track them more efficiently by having multiple colors localized to the nucleus. And we've also been more recently using this model to transplant human cancer cells like metastatic melanoma or neuroblastoma back into the embryonic microenvironment where we have single cell resolution, and ask the question, do these cells have any memory for that embryonic microenvironment, and can we potentially reprogram them with these powerful signals that are there? So going back to this discrete stream of the neural crest, we know a little bit about the molecular cues early when the cells are shaped or sculpted into an initial stream. And that's work from many different labs, including Marianne Bronner and Laura Gamel's group. And around the neural tube, you have factors in the neural tube that are expressed and then thought to be secreted locally into the microenvironment that restrict neural crest cells from going in an uncontrolled manner and kind of funneling towards or down this stream here. But what's challenging to me, and has been sort of the part of the questions we've been asking for the last couple of years now, is what about the persistence over long distances? Because this is a local effect, why don't they go into these areas as well too? So what keeps them directed, and why don't they stray farther out from the vicinity that's just about 100 microns from the neural tube, or the range of those potential inhibitory factors? Yeah.
In the left-hand picture, they do seem to spread out quite a bit as they move out.
Yeah, I'll tell you about that, because the front seems to be more exploratory. They've got phylopodia in lots of different directions, and then the more trailing cells, or the trailers, seem to be more bipolar and sort of locked onto the pathway. Okay, so today I want to tell you about our model for this, a cell-induced gradient model that we've been going back and forth between a computational model and experiment, and then more recent evidence for a molecular signature for the most invasive cells, and then finish with challenging our model, and then a couple of questions about how I think computational modeling can help us now move forward from where we're at right now. So just to give you a little bit of a background, the neural crests have a rich history in modeling, going back to around 2007, and there Don Newgreen, who works in the gut, and Kerry Simpson in Australia put forth a model about frontal expansion of neural crest cells that populate the gut. And what they observed from experiments and then in looking at modeling was that you have this frontal population of cells here that expand by proliferating and then push through the gut this way and populate it by both cell movement and proliferation. On the other hand, in contrast, Roberto Mayor's group at UCL here has put forth a model for neural crest cell migration where you have cell migration more of a sheet of cells that occurs in xenopus and zebrafish to some extent early on before the cells are migrating much more as individuals. And Roberto suggested that contact inhibition drives this migration, where cells will break free from the front edge here and then leave a gap open between them, and then which the trailing cell will fill into. So contact inhibition as a mechanism. So to look at that in our system, in the cranial neural crest, we investigated this proliferation aspect a little bit further by doing time-lapse imaging and cell cycle analysis. And what we found is during the migration, there's very few cells that are actually proliferating. And they're on about a 16-hour window of the total 24 hours it takes them to get to the target site and populate it. And if you look at the number of divisions, you really see that it's a small percentage of the cells that are dividing. So this doesn't seem to be a frontal expansion of what's going on. And we confirmed that through photoconversion. So we loaded our neural crest, pre-migratory neural crest cells with a photoconvertible dye called KICGR, and we photoconverted just the lead sort of 20% of the stream here early on, and then waited 24 hours till they filled the target site. And we saw a dramatic expansion of those cells in the target site. And when we counted that, we saw, sure enough, if you look at that front, they go from small numbers to large numbers after they enter into that target site. But during the migration, there's not much proliferation going on. So in other words, the cells are not wasting energy to stop and divide. They're very actively migrating. And the ones that do divide are doing it fairly rapidly, but keeping their spatial position. So look at the cell cycle, how it changes. It's down to 4.5 hours after they enter into the target site. And then some of the trailing cells also decrease their cell cycle and exit into G0 to start a differentiation process. So instead of frontal expansion, it looks like we don't have this large front here that's expanding. But this has actually changed a little bit because of experiments from Heather Young's group and Hideki Enomoto, where they've generated kick-GR mice and shown that the lead cells that are advancing into the open portions of the gut actually slow down and proliferate and have less directionality and start to colonize the tissue. And if you look at the trailing cells behind them, they leapfrog those cells because they maintain their direction and their speeds. They go right past them, and then they become the ones that populate up ahead and then go through this expansion and circularization there. And so it's a refinement of that original model from Don, then, that you have a frontal expansion, but you also have this leapfrogging event by the population that comes in from behind, populates that area, proliferates, and then really colonizes it. Okay? So going back to our model, then, we went back to the photoconversion experiments and saw if we look at these cells in the front, they pretty much maintain their spatial order over the migration. So we do not have leapfrogging going on in the cranial neural crest. So neither a frontal expansion or a leapfrogging process. But then what about this contact inhibition of locomotion? Well, when we look at the cells, here's a cell right here that's reaching out. It's trying to make a decision on where to go. We see a lot more contact guidance. So a cell will reach out and then pull it back and then follow that cell it's made that contact with. So instead of cells bumping into one another and then imparting a velocity or a direction with each other, in what we're observing, we see a lot more of cell-cell contact and then moving in the direction of that contact, either by pulling back the filopodium and then going, or the filopodium kind of tracking the trailing edge of that cell. Okay, so it seems like much, oh, sorry.
But isn't Roberto proposing a co-attraction mechanism as well?
Yeah, more recently he's proposing a co-attraction mechanism, but that wouldn't fit right here. Remember, because the co-attraction is a local chemoattractant around the entire periphery of the cell. So how does the trailing cell know just to stick its filopodium right at the back end of that cell, right? It's quite different from Dictio, where Carol Parent has shown a localized secretion of cyclic AMP. You know, so we're struggling with that a little bit, but that's a great point, and I'll bring that up a little later.
What does the matrix look like?
Yeah, so the matrix is here, and we think it's dense, but it's difficult to see. You can see it at EM levels. Earlier on, cases looked by second harmonic generation and not seen very much, but it may be because the fibers are very thin. But by EM, you can see it there, and you can see that they're also crawling within loosely connected mesenchymal tissue or mesoderm that's populating this portion of the stream. So they've got to fight through that as well, too. That's part of the reason why I'm here about thinking about cell mechanics, and I'll get to that towards the end.
I think a good method to image fiber orientation would be polarization. And
absolutely. It would be really cool to see whether there is fiber orientation there, or whether the cells that are these lead cells that I'll tell you about have special properties are altering the matrix to make it easier for the others to follow. Okay? You're going to almost my conclusions, so if you have to step out and get coffee, I'll understand. So there's also, this is not happening on a static domain. The domain is growing while they're migrating. So if you look at, this is the same magnification of tissue sections over the development or over the migratory period. Look at how small this is. This is where the cells are starting and coursing out. And this is at the end when they're getting into the target site. So the tissue's bigger. We then looked at lots of different sections and with some help from some quantitative people then plotted that, and it looks like it's increasing, and then it really sort of peters out when the facial processes come together and fuse. But you really have these cells actively migrating on a moving domain, and if you label the domain and you label the cells are actually traveling a little faster than the domain is moving. Okay, so it's the person on the moving walkway at the airport that's moving a little bit faster than the neighbor that's just standing on the moving walkway. Now, from a molecular perspective, we've got some evidence. If you remember some of those cues earlier on were the semaphorins, which are also axon guidance cues, and neuropilins being involved. We looked at one of the other ligands for neuropilin 1, which is vascular endothelial growth factor, and we found expression of it in the surface ectoderm directly overlying the migratory route with the neural crest cells underneath it. And its spatiotemporal dynamics were really cool because you could see as the cells were going farther down to the target site, so was the expression of VEGF, and it became high in the target site. These are the second branchial arch where they're headed, and we had high expression of VEGF. So we naturally asked, is it a chemoattractant for the neural crest cells? So we placed VEGF-expressing cells into the embryo, into a neural crest cell-free zone, and we could divert some of those cells locally to encounter that source, and also divert cells from above in the other stream to come down and encounter that source. And if you see it at sort of low resolution here by di-I and di-O labeling, then you can see some of these cells within range of the VEGF source will divert and meander and mingle around here, while some of the other cells continue on their way. So we formulated this model then where we hypothesized that VEGF signals direct neural crest cells into the branchial arch targets. Now remember, VEGF is expressed ubiquitously in the surface ectoderm. We think it's locally secreted and bound. It doesn't diffuse very far. But it's everywhere. It's not spatially laid out, so it's just in the neural crest cell free zones or in the stereotypical pathway. So we think what's going on, there's this dilution due to growth and consumption by the cells. And at this point, we asked Hans this question: how can a computational model help us understand neural crest cell chemotaxis on a growing migratory domain? And I naturally turned to a good colleague friend of mine, Philip Maney, and asked Philip, is there anyone that might be interested in this? So together with Ruth Baker and a PhD student for the first couple of years, Louise Dyson, whose work I'll show you, and then more recently Linus Schumacher, who's just finishing, paired them up with a more senior developmental biologist in the lab, Beck McLennan, and a research technician, Jessica Teddy, and then Jason Morrison, and embedded them sort of for a few weeks, let us talk about the modeling, and what we came up with was a two-dimensional domain on which is an agent-based model where we have individual agents here that read out VEGF concentration in the migratory domain. So the chemoattractant looks like spatially there's a short-range diffusion, there's consumption, and a production. Okay? And this is on a growing migratory domain that's embedded in this. And when Louise ran the first simulations, she said, this is great. You get the cells coming out and coursing out, but what happens is they are looking for VEGF. They consume it as well, too. It's diluted due to growth, and you leave a bunch of cells back at the entrance to the origin where they came in. And we said, yeah, but that doesn't happen. And she said, yes.
So what is the color red? Is that?
That's the VEGF concentration. Yeah. So she told us that, and we're like, this doesn't sound right. So they said, you need two subpopulations of cells. You need lead cells that will read out the VEGF, and you'll need trailing cells that get their cues or instructions on where to go from the lead cells that are reading the VEGF. And we thought, that's interesting. So we revised the model then to say that these cells in the front are leaders reading out VEGF, and the trailer cells, a separate subpopulation within the stream, follow the lead cell instructions. Okay? So the next thing was on us, because what we would do is we would work in isolation, where we would meet in Oxford or meet in Kansas City and then go away for 6 months, and they do modeling, we do experiments. So then we went back to the lab and said, is there a molecular correlate that would identify lead cells versus trailing cells more uniquely? Yes?
What's the percentage between leading cells and...
Yeah, we'll get to that. It's a great question, right? So what we initially did was just look at that lead sort of 20 to 30% of the stream and cut the cells out by laser capture microdissection and compared their expression for about 100 genes by RT-qPCR with the trailing cells. And we found, sure enough, there's certainly a difference between the lead and the trailing cell gene expression profiles. The lead cells have high expression of MMPs, they have high expression of certain integrins, they have low expression of certain cell adhesion molecules, and they have high expression of the receptors for VEGF. So we thought, this is really cool. The trailing cells have high expression of cell adhesion molecules, they've downregulated the MMPs, they've downregulated certain integrins, so this looks pretty neat. So then we went back and thought, this is a cell-induced gradient model then, when it decides to come back, where you have a lead cell population here that is reading out the VEGF, and it's diluted the growth and consumption, and you have a separate trailing population here. So the question then becomes, how do cells interpret the signals? How many cells do we need? Like this gentleman is asking. How many cells really need to interpret those cues, and how many are leaders? So I want to talk to you about evidence for a separate population of cells that have a unique molecular expression profile at the front that we're calling trailblazers. So what we next did is go back in the lab and split up the stream into certain subregions here, 8 of them here, at a couple of different time points, and look very carefully at what the expression profile is in each of these subregions within the stream. Okay? And what we found is that the gene expression profiles have a lot of spatial variability. If you do the hierarchical clustering, the first population in the front here, the first 10% of the stream, roughly in subregion 1, have a molecular expression profile that's somewhat similar to subregions 2 and 3, but 4 and 5 seem very unique, and then the trailing cells, or the cells that just have recently emerged into the microenvironment, have a much more unique expression profile. And when we looked at this a little bit closer, we wanted to confirm that expression. This is typical in situ hybridization, where you cannot resolve borders between gene expression very well. But there's another emerging technique called hybridization chain reaction, which is a fluorescence-based technique, which will allow us to see multiple expression patterns in the embryo. And so we've been exploiting this together with combining with the immunolabeling. So here are the neural crest cells in a typical section from the dorsal neural tube along the migratory pathway, labeled with H and K1. And here are 2 of the genes that showed up, BAMBI and HAND2. And you can see expression of HAND2 is high, closer to when the cells are entering the target site. BAMBI has a little peak here and a peak in the middle. And to analyze this further, what we can do is, with the help of some quantitative analysis, is run a line or an area all along that migratory pathway and do a fluorescence intensity analysis and normalize it versus the distance along the neural crest pathway, and really see that peak at the front in HAND2 or that small peak in the front by BAMBI, and then especially this peak in the middle region there. So that's the way we're sort of breaking down some of these expression patterns. So we have high expression of some genes, but we have spatial variability or regional diversity of expression depending on where you're at in the stream. But the cells in the front in this subregion 1 seem to have a very unique pattern. So perhaps only some cells need to interpret the guidance signals. So then we went back to Philip's group and said, well, what happens if we start to eliminate some of those lead cells? What happens to the stream? How many leaders do you really need to do this? This is a really difficult experiment to do. We'd have to go in and photoablate some of those leaders, and that's challenging because you're heating up the local microenvironment. You don't know whether you're altering the expression profile of a cell or its neighbor. But in silico, this is much more easier to do. So we went back, and then Linus now came on board and said, I'm going to test whether very few neural crest cells interpret guidance cues and instruct others to follow. So what Linus did was he ran this experiment for having a changing mean leader fraction, and you're seeing a decrease from 1, where everyone's a leader in orange here, all the way down to 5% leaders here in orange. And what you can see when you plot mean leader fraction versus distance along the migratory route is, this is really sort of the best, simulation of proper migration. A small number of leaders that make it to the target site, and a good density of others that are following her, versus everyone being a leader, because everyone being a leader is searching for VEGF, and they just get stuck here because everyone's looking for VEGF and consuming it, and yet the signal's going that way. Yes?
So in the computation, you see a kind of envelope around the front of leader cells. Do you see anything like that in the experiments?
Yeah, I'd have to say that it may be, you kind of see more of a widening of the front, and it may be that they're also sampling that local inhibitory factor too, and playing with just how wide they can go, and in response to that persistence factor or chemoattractant. Here, we don't have anything except a no flux boundary condition on either side, and so we're kind of, limited in having an inhibitory factor shape that as well too. Yes?
What's the evidence for VEGF consumption?
In our hands, we don't know. That's a really challenging question, right? And there are in other migratory cell populations, but at least in the neural crest, we haven't even shown a binding event going on. That's a challenging experiment to do in vivo with a FRET sensor or something. But we don't know what the consumption of the VEGF is either. Yeah. Yes, I'll take that one first. In the back.
Is there like a role change where you see new leaders coming in and old leaders dropping back?
Yeah, there's a plasticity there that I'll get to in the end, where you can transplant trailing cells into the lead position, and what happens is the original model now, because remember, these lead cells are looking for the chemoattractant, and these cells are looking for leaders. So when the model predicts that if you take those trailing cells and you put them into the lead, they're going to be looking for leaders and not finding anyone. They just stall. But the biology, when we did that experiment, we put the trailing cells into the front, and then bam, away they went. So the microenvironment is driving this gene expression profile, and there's plasticity in the neural crest cells. They're not hardwired with this profile. And then so we think what's going on is then, and profiling showed that they're upregulating all the genes of a lead cell phenotype, and then away they go. So then we had to revise the model that if there's a plasticity there, depending on your local VEGF. Yes?
So, to be clear, so you don't think there's a preset VEGF gradient, that it's rather flat, and it's the leader cells through consumption that somehow create this gradient. So I was wondering if say that's true, if you were, by tracking individual cells, if you saw, I would imagine that one consequence of this would be that the little cells are not actually as persistent in the migratory patterns as the followers. Because no, there's no gradient. At least the very leader cells, there's no actual gradient.
Mm-hmm.
The gradient is being generated, according to what you're saying, by those leader cells. Do you
Yeah.
get that?
We see that those lead cells, and we haven't backed up to when they first enter the microenvironment. That's where we're trying to go. But we do see them moving slower and more directed.
More directed.
Yeah.
That may not be completely, let's say, compatible with this notion that leader cells would be actually generating effectively this gradient.
Right. So I think it's important to go back and look when they first enter the microenvironment, and they're first encountering VEGF, is first of all, when do they start upregulating these genes that they're going to become leaders, and how are they altering the chemoattractant profile? There may be other signals out there as well, too, that might be graded. We don't know, because neural crest cells can respond to graded signals we know in the trunk. So I think you bring up a good point, and we'll see how that plays out a little bit earlier. We just those numbers about slower speed and better direction seem to be sort of mid-route to end, where we really sort of can track them for a longer time.
Could you disconnect leading cells from trailing cells, and then, similar to what Darren Gilmore's done, lateral line and see if they're turning around?
Yeah, so the only thing that we've done is we've stopped the trailing cells from coming out by ablating them after the leaders have come out a little bit, right? And what we expected from the model is they would just keep continuing well, we expected that they would just all go to the target site. The model told us that no, that's not right, because some of them are following VEGF, but it's moving quickly, and some of them get left behind, so they're distributed along the migratory route. And that's exactly what happens in the experiment. So I think we're kind of at the same point as Darren would argue, that maybe just a few cells, as you know, in lateral line are enough to guide the whole cluster, and a few cells in the trailing edge that are sequestering the ligand underneath it to create a gradient there. Maybe you only need that, and cells in the middle might be responding to a completely different signal, as Dennis might be suggesting that there's other signals out there. Okay, so can a very few cells guide the stream, and how efficient? Here's the 5% leader fraction versus the 50% leader fraction. And you can see it seems it's much more efficient to move the stream when you have just a few of these cells that are out in the lead there. Okay? So that told us that maybe we need to look for fewer leaders. This is a difficult experiment as well, too. So we went and looked at the single cell level now, and single cell gene profiling of these 100 genes by cutting out the most invasive cells at the front. We'd only get two or three cells per embryo, and when we did that, we were able to get good numbers here and good resolution of just those 2 or 3 cells at the front. And what we found was that there's a consistent set of genes expressed by those lead cells, about 16 out of the 100 genes we're looking at, that were consistently and highly expressed at this mid-route, and then also when they were entering into the target site. And when you look at those set of genes, you see high expression of some chemokine receptors, a desmoplankin here, NED9 that's been argued as a factor in cancer cell invasion, and our NRP1, our neuropilin-1 for VEGF, and similar genes here, so much so that we have now made a list of the 16 genes that we are calling a trailblazer molecular signature. And these cells are just the ones that are narrowly confined to the migratory front. They're the most invasive. And so we think this is the signature of the most invasive cells. Now, this is sort of a plug for single cell analysis, because when we compared our single cell analysis versus averaging of the subpopulations by cutting up subregions 1 through 10, this is what we found. So we typically look at violin plots, which show the expression here as a function of, at this point, stage 13 and stage 15 in green. But the width of the violin plot tells you really cell number, and the length along here tells you expression level. So a gene like Bambi at both of these phases of migration was high in a large number of cells. And the same thing for a chemokine receptor, CXCR1. But if you look at a couple of other genes we analyzed, like HAN2 and SOX10, you can see that there was super high expression, but in a very small number of cells, that when we averaged it was just driving up that expression level. But it really was falsely indicating that HAN2 is expressed in a large subset of the population, but it's only a few cells. HAN2 is more related to neural crest differentiation, and that's suggesting to us that during the migration, they're actually regulating their differentiation earlier than when they actually get into the target site and see some of those signals. There's a subset of cells where their plasticity is changing en route. Okay? So going back to the model then, we revised it again to suggest that we have these lead cells very close at the migratory front here with a distinct molecular signature that we are calling trailblazers, and we have a trailing population here. And so now we wanted to next challenge the model. What happens if we alter the VEGF signals, and do we change neural crest cell behaviors and gene expression? So we thought about these two experiments with Philip's group about placing an ectopic source of VEGF adjacent to the stream, introduced only to the trailing cells, because we know before that it's chemoattractant for the leaders. But what about the trailers? Are they hardwired for that? And what if we put VEGF directly into the migratory stream in the trailing population? Would they turn around? Would they respond to it at all? And so what happens when you put that ectopic VEGF in, you break the stream. The cells that are coursing down this way will reverse direction and coalesce alongside this portion of the ectopic source. And these cells encounter it and then can move through that or either stop and coalesce there for a while. And the model simulation where Linus put the source of VEGF in the trailing population after a time showed that you do get this coalescence of cells back here around that source as well too, because when you put the source in, remember there's a plasticity built into these cells now because of the experiments we had from trailing to lead that showed that the leaders could trundle off. If they see VEGF, they can respond to it. Okay? So we know we can alter the stream and the behaviors. What about their expression profile? So what we did next was look very closely at the cells that had encountered the VEGF and ask about their gene expression. So did trailing cells in the presence of VEGF upregulate genes of this trailblazer signature or not, right? And what we found is for the most part, when we placed a ectopic source within the stream, for the most part they seemed pretty similar to trailing cells. They didn't alter their genotype significantly. However, there were 9 out of 96 genes that did, and 4 of them were in the trailblazer signature, suggesting that VEGF can induce the expression of these particular genes, some of which are in the trailblazer signature, and in the trailing population we can convert them, and these are part of the trailblazer signature. So it could be, Dennis, that there's other microenvironmental signals out there that they're responding to that are driving the full aspect of this signature, but VEGF, at least presented to the trailers, can upregulate these genes. So to summarize so far, I've talked about the cell-induced gradient model where lead cells respond to VEGF and guide the trailers, and given evidence for a trailblazer signature and challenged our model with VEGF and gene expression profile. But now I'm to the point of, now I need to come here and ask the computational modeling folks 2 important questions that we're on to now. So what's the function of the trailblazer signature genes? How important are they to the stream? Okay, what's their function? What can we drill down in mechanism and see what's really important about these genes? We know from preliminary work that we can overexpress 1 or 2 of these in the trailing population and make them more exploratory. But why is it that you have some genes like aquaporin 1, you have a host of chemokine receptors, you have some Fs in here, and you have a very high expression of a desmosome? What are they doing? And just to give you a little bit of a sample of that, one of the genes that we're looking at is aquaporin 1, and I'd really appreciate anyone that has some thoughts about this and maybe bringing in some cell mechanics or some fluid mechanics into this, because aquaporin regulates the water channels in a cell, and so if you have local water channels opening up in this cell, it's thought that maybe, and there's some evidence from glioma cells, that they adjust their cell volume by regulating those water channels. So do you have this super high expression in only the most invasive cells of aquaporin 1 because they're regulating a water channel to change their local morphology to squeeze through the matrix or crawl over other cells? I don't know. But with the help of a couple of colleagues who are the epinephrine signaling people, David Wilkinson in London and Phil Soriano in New York, they pointed me back to a paper in 2000 that said, but there's some evidence that aquaporins can form a stable complex with epinephrines. And the epinephrine binding with those aquaporins is thought to, and this is the last, almost the last sentence of the discussion in this paper, thought that it might be that this aquaporins and anti-exchangers may also function in the growth cone to help integrate pathfinding information where you have these epinephrine receptors localized. And so maybe the growth cone can respond to highly localized changes in water transport or pH and become destabilized, leading to repulsion. So if it can lead to repulsion, can it lead to protrusion as well too? So I need help thinking about or a computational model that would help us understand how local cell shape mechanics that's mediated by aquaporins lead to cytoskeletal changes in response to chemotactic signals. Okay? And in the last couple minutes, I want to tell you about the second question that we're interested in, which is the one that I wrote on the board, which is what is the mechanism of communication between leaders to trailers? So that's our second important question that's come out from the model predictions and experiment. So here, I think it's an open question. I think there's at least 3 different things going on here. There could be a cellular debris trail, or there could be cell-to-cell contact between leaders and trailers, or it could be a secreted factor from lead cells to alter the matrix or even attract the lead cells, right? Especially if there are a lot of these chemokine receptors there. Now let me just touch on some preliminary thoughts and food for thought for discussion over tea. What about a breadcrumb trail or a debris trail? Is there a debris trail being left by that lead cell that this neighboring cell is picking up on, right? So what we're in the process of doing is taking a multicolor, multispectral approach, is to label the neural crest cells, label the leaders preferentially with a couple of different colors, and then transplant them into a host embryo where the trailing cells are labeled with a couple of different colors, and see if we've got exchange of things between cells. Now, I thought this was kind of a fishing expedition and difficult to do from a microscopy point of view until my scientific director pointed me to a very recent paper that's showing that neutrophil trails guide influenza-specific CD8 positive T cells by leaving a pathway of debris of a chemokine ligand, CXCL12. So we've got a lot of chemokines, if you remember, as part of the Trailblazer signature. Maybe that ligand is being produced by a lead cell, and that debris from there is being left in the matrix, and then that trailing cell is picking up on that. I don't know. Okay? What about cell-cell contact that I mentioned too? So if you look at a little bit faster dynamics, here are a couple of neighboring neural crest cells extending lamellipodia and filopodia, and a branch between them, which looks kind of interesting because it looks like it's sustained. And we kind of got curious by that a couple of years ago, and it turned out by using a photoconvertible reporter, we were able to photoconvert one of the cells. This was a dividing cell that had just divided, and we photoconverted one of the progeny. And then after time, we saw the cytoplasmic transfer from this cell into that neighboring progeny as far as 120 minutes after the division process. And so that was really cool to us because it suggested, there's a lot of dynamics going on here. And if you watch that in time, you can see the photoconversion, and then the neighboring cells start to go from green to orange, suggesting that there's cytoplasmic transfer, and we've done a little bit more measurements on that. So the neural crest cells are definitely communicating by touch. Our preliminary measurements are showing that those contacts are filopodia to filopodia, or filopodia to cell body, or cell body to cell body. And there's some dynamics to the contacts that looks like it's bimodal. Either you have these really long-lasting contacts or very short-lasting contacts on the order of seconds or a few minutes versus much longer. So it may be that as they're traveling, they're poking one another to get a sampling of the neighborhood, but a long-lasting contact might be enough to alter the transcription in that cell or pass message to that cell of some sort. So it would be really neat to follow up that cell-cell contact process and whether there is this contact from a lead cell to a trailer that is then passed along to other cells from that trailer. Okay, so my question is then, how can a computational model help us rule out some of these hypothetical cell communication mechanisms. We've got at least 3 up there. Can we at least rule 1 or 2 of them out in connection with a couple of experiments that we can run? So is there more of a sort of a general model about cell communication and that we can adopt that would help us sort of rule out a couple of these mechanisms? Because to go down each one of those pathways, and I should say that in a brief comment before I finish, if you look at the list of 16 genes, what my biological colleagues ask me is, why don't you do RNA-seq so that you can get more genes and get a sample of what more, expand the signaling pathways? And when I talk to my computational modeling colleagues, they say, what's the function of that gene? Right? So, it's a disconnect there. So doing one gene is going to take us 2 years in the laboratory to do. Doing a couple of them in a combinatorial manner is possible, but it takes time, right? So if I can go back to these general mechanisms of cell contact or debris trail in a general sense, maybe I can at least rule out some things. So I wanted to just close there and thank Philip and his group and Louise and Linus for a wonderful collaboration, and Rebecca, Jessica, and Jason Morrison. And I'll stop there and take any questions. So thank you. Yeah.
So, directly to your last question, okay? I mean, between the beautiful developmental work you're doing and the computational model, there are many other possibilities, okay? So one possibility would be a reconstituted assay where you put the whole system into a more controlled environment. Like, for example, we have seen yesterday the cylindrical zebrafish, okay? To rule out that the curvature is important. So I wonder, can you actually put this into something like a patterned environment? And then actually the computational model become more helpful, because then you can actually check hypotheses on the role of geometry.
Right. So we wanted to ask whether this lead trailing gene expression profile is sustained in culture, because if you take a neural tube out of the embryo, you put it in culture into a 3D matrix, the cells will invade everywhere. When we profiled the leaders versus the trailers, the complete expression profile is lost. You don't have any of these trailblazer genes, and then even when you put in a VEGF source into that, they will only upregulate 1 or 2 of these trailblazer genes. No one has been able to recapitulate this stream pattern by putting in potentially inhibitors and a permissive factor, but maybe we're learning more about inhibitors now from profiling. It might be that we're able to then put into a patterned stripe assay, at least, some inhibitors and then a ubiquitous layer of VEGF concentration, and really get at this question that Dennis was asking, how is this gradient being formed by the cells? Is that sort of more what you're getting at?
I mean, I was more thinking about some kind of channel,
Mm-hmm.
and maybe some kind of transplantation experiment, like Keith has been talking about. You then transfer cells from one side of the channel to the other, something like this.
Yeah. And what would the advantage of that you're saying? I mean,
Because I think then it's easier to apply the computational model.
So you could play with concentration gradients over time and stuff like that.
Mm-hmm.
It's more quantitative.
Yeah, that may be important, because if you see those other chemokine receptors there, it suggests there's other microenvironmental signals, or the cells themselves are making the ligand, right? So VEGF's not the only one. If you knock the VEGF receptor down, the neuropilin-1 receptor down, they actually make it partway down the trail, but don't enter the target site at all. So there's other players there that might help us, because I didn't say, that's I guess part of that one slide, which was the last bullet point, was what if there are multiple secreted factors? How is the whole population responding to it? And Carl Philip was leading to that with the lateral line. They're at the same point now, because they've separated the cluster and seen different phenomena going on. So it's not just CXCR4 and CXCL12 guiding it. There's other multiple signals that are keeping the cluster together and guiding them too. So maybe by playing with the gradients in culture, we might, and we'd have to sort of, you know, my only worry is that you lose its expression profile from the in vivo scenario, you know? So if you can give me an assay like that I can transplant into the embryo.
The behavior of the model, and especially the dependence on the concentrations, on the numbers of leader cells, seems to be very much dependent on the rate of consumption of the VEGF. So, but I understood also it's not that clear whether the cells actually consume VEGF. So have you tried varying the VEGF consumption rate in the model and see how this changes the behavior of the model?
Yeah, and I'm sorry I don't remember the result, but I think that's something that Linus has looked at, but I don't remember the result. And I can ask him and get an answer for you later.
Okay.
But I think it's important. That's just a difficult experiment for us. And we don't have biochemistry background to do it, but we could collaborate with someone that would think about it.
Would there be alternative mechanisms that give the same behavior?
With VEGF and
Well,
instead of the consumption of it?
Well, for example, I could imagine one model without consumption where the leader cells are following the VEGF gradient or generating a gradient, and that the follower cells would act according to a contact inhibition mechanism.
Yeah. Yeah, I mean, it could be because, as I was mentioning, if you knock down the receptor for VEGF, they make it a certain way down the route, but then don't get into the target site. So it's absolutely crucial for them to have this interaction between neuropilin and VEGF to enter into the target site. It's kind of like a lock and key. So there's something else that might be in there, but I don't think it's a repelling, I don't think there's a repelling factor like Bakhtiar talked about yesterday, perhaps, that's pushing the population, but there might be another signal that the leaders are at least responding to or creating themselves.
Question. I'm just wondering, what is the default behavior of cells in the absence of VEGF or some other signal? So, I guess, you know, like what's going on in your model? Like, what do the followers do if there's no leaders? They just sit there, or do they migrate at a certain speed, a certain direction, a certain amount of time?
Well, in the absence of VEGF or in the presence of VEGF?
Even in the absence of VEGF. So, like, given the explant results, it seems that neural crest cells just like to migrate. They like to migrate in fairly straight lines. So maybe it doesn't take a lot to tweak them in the right direction. So it seems like the assumptions of what cells do in the absence of a signal could influence the outcome of the simulations quite dramatically.
Yeah, I mean, in the presence of VEGF, the trailing cells in the model will look for VEGF because originally we thought they were looking just for leaders, but those transplant experiments where you put them ahead showed that they could really get into the target. And so it suggested that they can alter that based on VEGF. And the model also predicts that in the absence of VEGF, you downregulate that lead profile and you become a trailer. Okay? But in culture, I agree. They are chemokinetic. I don't know if they're very, if you look at their trajectories, they're kind of, they spread out to populate the dish if you put a neural tube explant down. But I don't know that there are very many directed trajectories where they're going super radially and whatnot. So there's a
density or something.
Yeah, I mean there's stuff in the matrix because you've got growth factors as part of whatever matrix that you use because we're just typically using a 3D matrigel or et cetera assay. So I mean, that's a good question. I mean, they have an inherent motility and invasion to them.
So in the simulations, if there's no leaders, what do the followers do?
Become leaders.
Okay.
Hi. We have this lovely kind of experimental system, but I was more interested in how easy would it be for you to probe some of the assumptions you make in your model about the domain growth? Because beyond it being logistic, and we have logistic, and you have the idea that the cells are moving faster than the domain. Growth is quite passive. It doesn't do a lot. It's just a stretching. And so would you be able to test if that's true?
Yeah, that's a hard experiment without affecting the embryo in other ways too.
So you were able to show that the domain was moving slower than the cells. Wouldn't you be able to, in some way, be able to do it? How did you do that?
By labeling the domain by just putting in a few specks of dye and labeling the pre-migratory neural crest cells with another color and watching the two dots.
So in your model, if you did that, so if you put two bits of dye adjacent in the y-axis, by the end of the simulation they would still be adjacent. So that's the assumption in your model. And that would have quite profound consequences in a density-dependent system. So I just wondered to what extent, if you put two bits of dye adjacent in your experimental system, at the end of development, how far have they drifted apart? Because that's quite a different type of growth.
Yeah, I can measure that. I could probably do that experiment.
Yeah, because people don't do that. They just always assume stretching.
We've tested in silico the density by actually looking at increasing or decreasing the mean liter fraction in terms of the density. And we seem to mimic these results that Simon Levin and Ian Cousin have from a few years ago in silico as well too, that if you have a larger population, you need fewer liters. And if you have a smaller population, you need more liters. And so that seemed to play out in silico in this model. We just haven't done the experiments to that. Because we can reduce the number of cells by ablation, but it's harder to put twice as many cells in without affecting a gene expression profile, because we've got to release their ECadherins, et cetera, and whatnot. But I like the first experiment. We could try that on growth.
Oh, yes. So you were mentioning at the last few slides about these possible mechanisms that you would like to rule out, or to somehow have a criteria to determine which one is the one that is actually happening or the most important.
Yeah, because I've just proposed 30 more years of work.
Yeah, that's a very nice thing. But also, at the very beginning of your talk, you mentioned the analogies that people have been kind of pointing out regarding models of migrations in ecosystems, for instance. And in that particular set of models, where people have devoted modeling these sort of motions, they have developed a number of agent-based models in which they incorporate the strategies to every agent. These strategies can be passed by a number of mechanisms between the members of the population, and then you adopt one or another strategy depending on the outcome of at the population level, for instance, right? So one of the cool things that you could do with your model is to implement a gene profile, and that would be your strategy for a trailblazer cell. And then and they could just communicate by debris trailing. But also, every time that you can establish a contact, you mix the genetic material, and then it will be somehow of equivalent to adopt a bit of that strategy, and then you could prove or rule out computationally, according to some parameter tuning, which one of the two mechanisms happens to become more relevant. And that's pretty straightforward to make.
Yeah, so we should talk over tea.
Yeah.
Because people are looking at me now and saying that they need tea. But you're right. That's exactly why I'm here and why I want to have tea. That's a great, yeah, thank you.